Good evening, Gaurav. Good evening. Good evening. Good evening, Pankaj. Good evening, Gautam. Congrats. I hope you availed your Amazon voucher. Good evening, Saima. Good evening, Tarun. Good evening, Akshay. Okay. Good evening, Savita. Okay, let's start today's quiz. That is what on the molecular biology or the fundamental processes. Okay, so continue, Preeti. Good evening. Good evening, Sumaya. Okay, in this particular molecular biology chapter we're gonna discuss 16 to 18 questions in the period of 1 hour. So please try to understand every question seems to be very simple, but questions are little confusing. Some questions are very straightforward and some questions are little confusing questions. Okay, so try to focus on every single question slowly and try to understand and analyze in a correct way and give the answers. Okay, and friends, there is another session, that is question paper prediction. Okay, so give me 2 more days time. I have prepared the data, still half data only I prepared it consuming lot of time. Most probably we will conduct on the day after tomorrow, where I will analyze all the 13 chapters. Out of 50 and 75 questions in the part B and part C, out of 125 questions, at least we will predict 100 questions, because prediction we have to do very carefully. Okay. Okay, friends. Good evening, all. Good evening. Let's start the quiz. Okay. So let's say yes, we will start it. Let's say start. We will go it. Good evening, all. Good evening. Good evening. Please start. Say start. We will go it. Please say start. Yes, perfect. Okay. So friends, here you go. The question number 1, 30 seconds. Yes, start with the question number 1. Question number 1. Class 1 DNA topoisomerases are. Class 1 DNA topoisomerases are. Your time starts now. 10 seconds more. 5 seconds more. 5 seconds more. Stop. Okay, so friends, we know that class 1 DNA topoisomerases are ATP independent. Correct na? Okay, now everyone tell me, friends, I'm just asking, class 1 DNA topoisomerases are going to cleave the single strand or the double strand? We also know that they are ATP independent and they are obviously going to cut the single strand. Do not forget this particular point. So you know, right, topoisomerases are required for removing the coiling or releasing the stress from the DNA. Hence the correct answer will be B. Correct answer will be B. Okay, let's ready for the question number 2. Here you go. 30 seconds. Your time starts now. Question number 2. Oh, I'm so sorry. Question number 2. Question number 2. 15 seconds more. Quinolones and coumarins are the classes of antibiotic drugs. They are the target of. 10 seconds more. 5 seconds more. Stop. Give your answer. Gautam said, yeah. What about others? Fast. Good. Very good. Quinolones are basically acting on what, friends? Their ultimate target is what? Bacterial gyrase. Very good. Fantastic. Everyone has given the correct answer. See, always remember the coumarins have a high affinity for the bacterial gyrases. They compete with the ATP for the binding site of the B subunit of this enzyme and inhibit their particular action. Very, very simple. So remember the answer will be A. Good. Your time starts now. Get ready for the question number 3. Here you go. Yes, think and answer. Your time starts now. 1 minute. Your time starts now. Forty seconds more. 40 seconds more. 30 seconds more. Oh, Gautam, okay. 20 seconds more. 10 seconds more. Stop. Good. The correct answer will be A. The question says that the priming process during the initiation of DNA synthesis involves. So we all know very well, friends. Suppose example, this is a one nucleotide. So nucleotide contain sugar, a base, then followed by the phosphate group. This is the first nucleotide. Now let's take the second nucleotide. Same, the second nucleotide also contain sugar, then followed by the nitrogenous bases. Same here, then followed by the phosphate group. Now you all know very well the first nucleotide at the 3' group, 3' carbon, it contains OH group, which is the hydroxyl group. Nucleophilically it attack here. Correct, na? So the nucleophilic attack by the 3' hydroxyl group of the RNA primer. Very good, very, very good, perfect. So the option A is the correct. Get ready for question number 4. Your time starts now. 30 seconds for this question. Please go. siRNA interferes at. Your time starts now. Twenty seconds more. 15 seconds more. 10 seconds more. Stop. What will be the answer? The answer will be B. Post-transcriptional level. Correct, na? So, siRNA. Always you know, right? The siRNAs are basically interfere the process of gene silencing. Okay, so mostly, friends, they are going to stop at which level? It is going to stop at the transcription level. So once the transcription is over, we are going to get what? mRNA. And we know very well the mRNA is not biologically active. So in order to make the mRNA biologically active, we will do these several processes like capping, polyadenylation, as well as the splicing. During that time, if we want to stop our gene not to express, what do you mean by that? If we don't want to allow our RNA not to give rise to the protein, if I want to stop the protein synthesis, if I want to allow our gene not to get expressed, so during that time I can silence it. So that is what? A post-transcriptional level. Hence the correct answer will be B. It's a direct question. Get ready for question number 5. Your time starts now. 60 seconds. Think and answer. Don't get confused. Most of the students will get confused with this question. It's a very logical question. 40 seconds more. 30 seconds more. 20 seconds more. 10 seconds more. Stop. So look into the question. What is this? A rho factor. So friends, we all know very well what is the major function of a rho factor or a rho protein. It is basically involved. Anyway, the answer will be A. It is actually involved in what process? It is involved in the termination of transcription. Friends, we all know very well there are two type of transcriptions are there. One we called as rho dependent termination as well as the rho independent termination. Rho independent means mRNA on its own it will come out with the help of a hairpin loop like structure. But when you look into the involvement of the rho factor, very important, rho factor ultimate goal is what? See, this is the DNA molecule. Everyone look into this. This is the DNA molecule which is coming out. RNA is coming out. Now all of you look into the picture. Still the RNA associated with the DNA here. Correct na? RNA still associated with the DNA. That link we have to break. How can we break this? Of course by using the ATP. So rho factor is a ATP dependent RNA stimulated helicase which can break down RNA DNA complex. Because if the RNA wants to come out completely, it has to break down from the DNA which is associated with one of the strand of the DNA. So that complex has to be broken. In order to break it, it requires the energy. Good, perfect. The correct answer will be A. Perfect. So get ready for the question number 6. Your time starts now. 30 seconds for this question also. Okay, I'm giving 40 seconds for this because reading takes time. 40 seconds. Correct. Correct dear, correct Shreya. Rho always attaches at the 5' end and it starts moving towards the hairpin loop like structure. Good. 20 seconds more. 20 seconds more. 10 seconds more. 10 seconds more. Stop. Stop. Question number 6. The answer will be A. What is the answer for the question number 6? The answer will be A. So friends, everyone look into the question. Some viruses never use the DNA to carry out protein synthesis and have only RNA as their genetic material. Now this would lend to the evidence to which theory? Which means RNA first hypothesis. Correct na? The viruses, most of the viruses, they do not have the genetic material as RNA. So that's why by using this RNA with the help of a reverse transcriptase enzyme, it is going to synthesize the DNA. So always remember RNA is their primary genetic material. So which means that the starting component in the virus is not the DNA. The starting component in the virus is the RNA. That's why we call it as a RNA first hypothesis. Good, perfect. The answer will be A. Please get ready for the question number 7. Your time starts now. 1 minute. Your time starts now. 1 minute. It's a very logical question. Please think and answer. 50 seconds more. 40 seconds more. 10 seconds more. Stop. Stop, 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 stop. So question number 7, the answer is C. Very good, very good. The question says that the pyrimidine dimers of mutation formed from thymine or cytosine bases in the DNA. See friends, we read in the classes also many times, when you heard the name of a pyrimidine dimer, what is the common point you have to remember? Pyrimidine dimers are basically caused by the exposure to the high intensity of the UV light. We have also studied that this particular pyrimidine dimers we generally removed by using the photoreactivation method. You can easily answer 1. Next, in double-stranded RNA, uracil dimers may also accumulate. You know, can anyone tell me what kind of this it is called as? Everyone remember, when the uracil can occur in the particular DNA, you commonly know that uracil is the base which is commonly entered into the DNA during the process of what friends? Deamination. So we have discussed many times very clearly, when the uracil introduced into the DNA wrongly, we will use a specialized enzyme. What is that specialized enzyme? We call it as uracil DNA glycosylase enzyme. This is the enzyme we basically use. So by using this enzymes, what we will do? We will remove that particular wrongly inserted uracil. By which mechanism? We call it as excision repair mechanism. Especially we call base excision repair, or BER. So the correct answer will be 1 and 4. Pyrimidine dimer can be removed by photoreactivation. Wrongly inserted bases like uracil can be removed by the excision repair. Hence the correct answer will be C. It's not nucleotide excision repair, it's basically base excision repair. Nucleotide excision repair, we will remove the bulky damages. Correct, na? Not the single bases actually. Correct, Shreya. Cytosine on deamination. Very, very good. Your time starts now. Get ready for the question number 8. Your time starts now. Yes. Here you go. Your time starts now. 1 minute. Which of the following describes most accurately how secondary structures in proteins are stabilized? It is all about the stability. Slowly, slowly analyze. We discussed many a times.
Fresh to home. Totally fresh.
30 seconds left. 30 seconds left. $20 seconds left. 10 seconds. Stop, 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 stop. Stop. Okay. See, which of the following describe the most accurately how secondary structure? If you can answer easily without looking the question also. Saima, how it is said here, huh? We discussed very clearly, right? Hydrogen bonds are the major forces that is involved in the secondary structure. But I have very clearly said it to you. Through hydrogen bond joining different parts of the peptide backbone. But you people has written that, some people has given the answer between different amino acid side chain. First of all, everyone answer in the classroom more than 10-15 times I told you. There is a no role of the side chain, or there is a no role of the R group in the secondary structure. R group never ever involved in the secondary structure, never ever. Only involved in the secondary structure is what? Hydrogen bonds. Where the hydrogen bonds are present, how they make the bond to become stable or the stronger? By forming the peptide backbone. I hope you all remember also. How does this particular peptide backbone is getting formed actually? Between amino group of the first amine, between the carboxyl group of the first amino acid with the amine group of the fourth amino acid. Correct? Now we read many times. I to I plus 4, then I plus 1 to I plus 5 amino acid, I plus 2 to I plus 6 amino acid. It is not between the amino acid, it is basically build the backbone. Hence the correct answer for this particular question will be option D. It's not the option C. Hope everyone understand very clearly. Okay, perfect. Okay, just a moment. Okay, get ready for the question number 9. Okay, here you go. Question number 9. Your time starts now. 1 minute. Twenty seconds more. Anyway, even though you don't know the answer, try it. 20 seconds more. Stop. At least anyone. Everyone has given the answer as C. Okay. Anyone given the answer as B? Okay, good, Megha. Good. The answer will be B. The main distinction between the origin of the two classes of small regulatory RNAs, siRNA and miRNA, can be best described. You have to look into the word best described. Okay. And if you look into the option A, Si originated within the cell cytoplasm. miRNAs originated from the cell genome. You have to remember, friends, miRNA is not completely derived from the cellular genome. No. miRNA is actually originated from the particular component of the cell. It is not completely originated from the cellular genome. Whereas siRNA originated predominantly from the exogenous double-stranded RNA. So this is the most accurate statement. I am not saying that option A is wrong, but I am saying that option B will be the best answer or the most suitable answer, because here option A suggests siRNA originated within the cell cytoplasm, but exactly from where? It's predominantly from the exogenous compartment, whereas mRNA predominantly from the cell. It may originate from any compartment of the cell, but it is from the cell, whereas siRNA predominantly from the exogenous source. The correct answer for the 9 will be B. I am saying to all of you, A also correct, but if they ask you a combination of statements, undoubtedly you can go for A and B. I can agree with that. Since it's a one statement you have to choose here, the most best described statement will be B. Keep it in your mind. But whomever given the A, I can say correct. It depends on the option that they given in the examination. Please choose it. If they said combination, undoubtedly you can go for the A and B. Okay, there is a no question number 10. The question number 11. Get ready for the question number 11. Question number 11. Yes, your time starts now. 1 minute 10 seconds. Question number 11. Question number 11. One minute 10 seconds. Shreya, exogenous means external gene sequence. Okay, someone has given the answer. 20 seconds more, 20 seconds more. Question number 11. B. 10 seconds more. Stop. Okay, the answer for the question number 11 is the B. Okay, we have discussed many times in the triple L. Suppose example, this is the triple L RNA. In the triple L RNA, there are 4 complementary sequences are there. Pair 1, 2, 3 and 4. We discussed very clearly. When 1 and 2 pair with each other, they can give rise to a pass signal. When 2 and 3 pair with each other, they are going to give rise to anti-termination signal. When 3 and 4 pairs with each other, they are going to give rise to the termination signal. If you remember this, you can easily answer the question. Now question says that when tryptophan levels are high in the cell region 2 of the triple L is blocked from pairing with region 3. This allows the pairing of region 3 and 4, leading to the formation of a row independent termination. So 3 and 4 are the termination signal. What would be the structure of triple L region in the E. coli cell when protein synthesis has been inhibited? What is happening? Obviously region 1 and 2 will pair, allowing the 3 and 2 pair, leading to a process called as attenuation. What is the attenuation? Attenuation is the gene regulatory mechanism. Friends, if you quickly recall, in the E. coli we read 2 types of regulation. One is the negative regulation, one is the positive regulation. In the E. coli, what is the negative regulation? That is the operator and repressor. In the E. coli, what is the positive regulation? CAP and cyclic AMP. Same way, in case of a tryptophan operon, what is the negative regulation? Same, operator and repressor only. In case of tryptophan, what is the positive regulation? The positive regulation only, we call it as attenuation. Simple point it is. Hence the correct answer for this is the B. Question number 11. Okay friends, get ready for the question number 12. This question you have to think and answer. Question number 12. Your time starts now. 1 minute. Go with the question number 12. I plus Z plus Y minus I minus plus minus. Your time starts now. Oh, this much fast everyone. 20 seconds more. Stop. Okay, look into the given question. In the strain that have a genotype, dash synthesis of beta galactosidase will be constitutive. So you have to understand a question. Suppose example, this particular inducer, what is this particular I gene? This is the one which is basically synthesizing the repressor. Repressor will come and attaches to what? Repressor will come and attaches to the operator. If operator is blocked, RNA polymerase enzyme, an example, let's say an example I plus, RNA polymerase enzyme cannot cross it. But here, what is the condition? All of you look into this. The condition is that there is a no production of the repressor. So we said that I minus. When there is a no production of repressor, what is happening? The operator will be free every time. Then RNA polymerase enzyme can easily cross the operator. It can synthesize LAC-Z, LAC-Y, LAC-A continuously. When the LAC-Z is continuously synthesizing, it can easily break down the lactose into glucose and galactose. So what kind of expression it is? We call it as a constitutive expression or continuous expression. We read in the classes very clearly different kinds of mutations we studied. Very, very good. The answer will be B. Fantastic. Okay, get ready for the question number 13, everyone. Okay, get ready for question number 13. One second. Okay, your time starts now. Your time starts now. Question number 13. Correct, Shreya. Whatever you said is right. Your time starts now. Forty seconds more. Please, please think and answer. Think and answer. Think and answer. It's a direct question. You don't need to think much. Stop. Okay, fantastic. It's a very direct question. The condition is where both the glucose is present as well as the lactose is also present. Very good. It's a straight question. Okay. You can see the explanation. It's a direct question. Get ready for the question number 14. Question number 14. Yes. 30 seconds. 30 seconds. Question number 14. No, Shreya, it's not C, dear, it's A. What is the mode of action of gentamycin? 30 seconds. Okay. 15 seconds more. 15 seconds more. Your time's stop. Please type, stop. No one. Only one mega. No. Question number 14. Okay, no, Bia. Where exactly on the 70s? Oh no. Question number 14, the answer will be D. 1 second. Question number 14, the answer will be D. Why? Whatever the people has chosen 70s ribosomal correct if I don't give the 30s ribosomal subunit. As all of you said that, see here, this is the 70s ribosomal consists of a 50s and consists of a 30s. So together we call 70s. There is no doubt about it. The gentamycin will come and act on the 70s, but where specifically? Will it acting on the large ribosomal subunit or will it acting on the small ribosomal subunit? It is specifically acting on the 30s ribosomal subunit. More specifically. So you might read only it is on the 70s, but it is not on the large subunit of 70s. It is on the small subunit of the 70s. The answer will be 30s. Shreya, you look into the explanation, dear. Unless you don't be hurry, Shreya. Okay, just relax, calm, and analyze the question carefully. You are only looking one or two statements and you are giving the answer. Slowly analyze the question. You look into the explanation once again and you can ask me once again. I will explain. Slowly look into that. So for the question number 14, the answer will be D. Slowly look into that, dear. Okay, next question number 15. Get ready. Question number 15. 1 minute. Here you go. Shreya, you look into that. Once again, I will help you. Your time starts now. 1 minute. No problem, you look into it. I'll help you once again if you don't understand. Question number 15. "40 seconds more. 40 seconds more. "30 seconds more. Friends, please think it. Gautam, everyone, Mrunalini, okay. Slowly, friends. Think, you're not thinking. Savita, how it is B, Savita, huh? What you are thinking? Tell me how it's B. Stop. Question number 15. The answer will be the D. Option D. Option D will be the answer. 2, 4, 1, 3, 5. Okay. Now everyone listen. What is the first step, friends? We discussed many a time. First, small ribosomal subunit will come. Correct, na? So first, small ribosomal subunit will come. Right? Then once the small ribosomal subunit will come, the primed small ribosomal subunit bind with the large ribosomal subunit. You know very well, once the small ribosomal subunit will come, even tRNA also bring the amino acid. Now this small ribosomal subunit associated with the large ribosomal subunit. So first one is what? Option 2, the small ribosomal subunit is located. So this is the first option. Then the primed small ribosomal subunit binds with the large ribosomal subunit. So 2 of 4. Now after what is happening, what is the most important step next? That is peptide bond formation. What is the ultimate goal of the protein synthesis? Translation, ultimate goal is what? Synthesis of protein. Now once the synthesis of protein occur, we know very well there is a process is called as what? Translocation. Moving the ribosome from one codon to the next codon. That is what do we call translocation, which can be mediated by a specific elongation factor we read. So immediately what is happening after this? The intact ribosome slides forward 3 bases to read a new codon. Then what is happening, friends? New tRNA has to come. New tRNA comes into the which site? Obviously to the A site. So elongation factor will bring the amino acid tRNA, amino acid plus tRNA to the A site. Hence the correct answer will be what? Option D. 2, 4, 1, 3, 5. Don't be hurry. Relax slowly. Slowly choose and answer. Get ready for the question number 16. Correct, dear. Elongation factor GGTP. That is in case of eukaryotes. In case of prokaryotes, I hope everyone remember, right? Elongation factor TUGTP molecule. We read it. Okay, get ready for the question number 16. Your time starts now. 1 minute. Do it slowly, Shreya. Don't be tensed. Question number 16. 1 minute. They're asking correct. 30 seconds more. Ten seconds more. Stop. Okay, look into the question, everyone. Okay, anyway, quickly give your answers. Savita, Saima, what's the answer? Sumaya, Bia. How dear, huh? The answer will be D. Many a times we read, right? All of you listen to me. What does the spliceosome is made up of? Spliceosome is made up of RNAs plus proteins. Correct, na? Spliceosomes involved in which process? They are involved in the process of splicing. We read different type of splicings. We read a process called as simple splicing. We read a process called as trans splicing. We read a process called as alternate splicing. We read many processes in the class. Now look into the option P. They are composed of RNA sequences that signal for the removal of intron. Ultimate goal of the spliceosome is what? Removal of the intron. Option P is absolutely true. Option Q, they recognize RNA sequence that signal for the removal of the introns. Of course true. I understand, don't confuse with option Q. Option Q says that they recognize the RNA sequence that removal for that code for the introns. See here, this is mRNA. mRNA contain exons also. These are all exons. And these parts are what? Intron. Now those introns only we have to remove by the spliceosome. Option Q is also correct. Then option R says that they can produce different mRNA molecules by splicing at alternate splicing, at alternate site. How many times we studied the best example for the alternate splicing? We are having 5 antibodies, but all the 5 antibodies are synthesized from what? One mRNA only, right? By how? Alternate splicing. In the classroom, we have discussed one very good example for the alternate splicing. I hope you remember. Sex determination in the Drosophila melanogaster, which contain the double-stranded RNA. Right? We read now. What is that? Alternate splicing. What is happening? Alternate splicing. Exons are skipped and introns are added. Correct? Now exons are skipped differently. I'm sorry. Introns get deleted and exons are skipped. Suppose example. Exon 1, exon 2, exon 3, exon 4. What happens? Exon 1, 2, 3 will add, 4 can skip. Otherwise, 1 can skip, 2, 3, 4 added. Otherwise, 1, 2, 3 skipped, 4 will be added. This is what splicing we read. Alternate splicing. That's what they have given in the option R. Obviously, option R also correct. Hence, the correct statement for correct answer for this question is option D. Hope everyone understand correctly. Yes, get ready for the question number 17. Your time starts now. Your time starts now. Question number 17. Slowly, friends, slowly think, analyze, think, analyze. Thirty seconds more. Ten seconds more. Okay. 5 seconds more. 5 seconds. Stop. Good. Okay. Who has given the correct answer? The question number 17, answer will be A. Okay, they are asking which is correct. RRF and EFG aid. See, for continuation of protein synthesis in bacteria, ribosome need to be released from the mRNA as well as dissociate into subunits, which means after the process of protein synthesis, large ribosome and small ribosome has to be separated from each other. That's the process. These processes do not occur spontaneously. Of course, every process can be mediated by a several type of enzyme. We discussed in the classroom very clearly. They need all these. RRF and EFG aid in the process. An intrinsic activity of ribosome and all uncharged tRNA are required. Then IF1 promotes the dissociation of ribosome. Now anyway, what is RRF? Ribosome releasing factor. So that is extremely important to dissociate the small ribosome and large ribosome. And what is EFG? Friends, EFG is the one which is involved in which process? Translocation process. Because whenever a tRNA, uncharged tRNA present on the P site, uncharged tRNA has to come to which site? E site. That can be mediated by what? EFG. If uncharged tRNA comes to the E site or exit site, then only ribosomes can be dissociated. Otherwise, ribosomes cannot be dissociated. So, ribosome recycling factor is compulsory. EFG is also compulsory. Option A is correct. Now, in the option B, they said that an intrinsic activity of ribosome and all uncharged tRNA are required. First of all, friends, ribosome do not have any intrinsic activity. Ribosome can always depends on the several other factors. Ribosome on its own, it cannot come out like RNA. It cannot do that. Now option B is wrong. IF1 promotes the dissociation of the ribosome. IF1 is not the major one. IF3 is the major one. Because see, this is a small ribosome. This is a large ribosome. Whenever IF3 with the and IF1, majorly IF3 present in the small ribosomal unit, large ribosomal cannot associated with the small ribosomal subunit in majorly in the presence of IF3. So IF1 alone cannot do it. Majorly IF3 and IF1. Hence B is also correct. Hence option A is correct. Option B is also correct. Hence the correct answer will be A. I hope everyone understand. Okay, get ready for the question number 18. You can read the explanations. Question number 18, 20 seconds. Friends, I know you all people will make mistake of this question. Think and answer. 20 seconds. $20 seconds. 10 seconds more. Five seconds more. Stop. Stop. Who has given the answer? See, the question asked is alpha amanitin inhibits. You know very well RNA polymerase 3 will show the sensitivity, but it cannot be inhibited by the alpha amanitin. What about RNA polymerase 1? No sensitivity. But what about RNA polymerase 2? It will show very high sensitivity. Since it is showing high sensitivity towards this alpha amanitin, it can easily inhibit. Do not think that Pol3 and Pol1 are inhibiting. Pol3 can show the sensitivity towards that, but the toxin cannot inhibit it. Pol1 cannot show the sensitivity, it cannot be inhibited by the alpha toxin. Simple question. Okay. Get ready for the question number 19. Your time starts now. 1 minute. Here you go. 1 minute. Ten seconds more. Stop. Stop, 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 stop. The answer for this particular question will be the B. What will happen immediately after the transcription? What will happen immediately after the transcription? See friends, once the mRNA gets synthesized, immediately what is going to happen? The 5' cap, that is the methyl 7 guanine cap will associated, then followed by the splicing occur, then followed by the polyadenylation takes place. So first one is what? A methylated guanine cap will be added to the 5' end of the transcript, right? Hence the correct answer will be B. Okay friends, that's all for now. In this chapter I have taken every concept to two questions, and all of you listen my statements. Tomorrow is the most important. Why it is more important? Because tomorrow we are going to cover cell signaling, we are going to cover cell communication, we are going to cover cancer, we are going to cover host pathogen interactions, we are going to cover immunology. So since it's a very broad concept, from every topic I will take 5 questions. So every topic I will take 5 questions. So minimum you will be having 22 to 25 questions. Okay, I'll make easy questions, medium questions, and very difficult questions also. Make sure that everyone has to answer at least out of 25 questions, at least keep a target of 17 to 18 questions correctly in the tomorrow class, which is more important for us, because unit 4 is the biggest, one of the biggest chapter in our CSIR syllabus, where you can expect nearly 6 part C questions, even 4 to 5 part B questions also. Hence we should not neglect this particular chapter of unit 4. Okay friends, that's all. I feel the discussion is a little useful to all of you. Let's see the today's winner. I'll meet you in the tomorrow class. Okay, thank you all, thank you so much. Bye bye. Thank you all, thank you.